Welcome to our Delmarva case study presentation. My name is Ashley Skinner from the University of Florida, and I am pleased to introduce to you two individuals that wear many hats in produce safety, extension, and research, but are here today for this discussion. Welcome, Michelle Danieluk, professor from the University of Florida, and Laura Strawn, associate professor at Virginia Tech.
Perfect. Thanks, Ashley. Laura, thrilled to be chatting with you today about the Delmarva Peninsula and some Salmonella Newport outbreaks that have happened there over time. Can you start by giving us a little bit of information about what the Delmarva is?
Yeah, so the Delmarva is, of course, a little bit of a nickname, right? Delaware, Maryland, and Virginia. It's a large peninsula on the East Coast. Only fitting, I guess, that I'm talking to you from Florida. We used to call it the baby Florida. Very similar. It's got the Atlantic Ocean on one side and the Chesapeake Bay. You can see on one of the pictures below, that's actually if you were traveling north through Virginia Beach, you cross about a 22-mile long bridge to get to the beginning of the Delmarva Peninsula. And it's about 170 miles long. At some points, it's only 12 miles wide. So it is this really cool collection of wildlife sanctuary, habitat, rural community, lots of agriculture. You can see crops, tomatoes, cucumbers, snap beans, potatoes. It's got great soil for growing things, a great climate because we get that temperate, the water kind of protects us from getting too cold. Lots of poultry as well. And we also have a really big community of seafood production as well. So the watermen. So it's a really neat place. I lived out there for about 6 years myself. So
perfect. And Laura, I'm going to ask you a little bit, we've talked about salmonella in our series before, but can you give us a little bit more information about salmonella and the type of disease it causes?
Yeah, so salmonella causes salmonellosis. Salmonella is typically a zoonotic pathogen, which basically just means that it's commonly found in animals or warm-blooded, cold-blooded animals. So you got reptiles, you got any of your typical mammals, right? Can be birds. And if someone were to, it's typically transmitted fecal orally. Like I said, animals, right? So if they deposit feces or poop, you got the poop behind me, which is always a nice visual. Then if you're handling something and you don't wash your hands, potentially then have contact with mouth, you can ingest it. Once it gets inside your body, that's where it's going to start to go to work and potentially make you sick. Typically attacks the elderly, young children, immunocompromised, pregnant women, can make you pretty sick. And the really icky thing about salmonella or salmonellosis is that it can cause a lot of long-term health effects, arthritis, things like that. So we really want to avoid it if all possible. So does that kind of give you
So Laura, you talked about salmonella being a zoonotic pathogen or coming primarily from animals, but we can find it in the environment a bunch too, right?
Yeah, and that's what makes salmonella challenging, right? Or that we spend a lot of time and attention on it. Salmonella, there have been published studies that have been where we can show it can be found in soil, water, I mentioned manures and feces. And a lot of times it's basically just kind of going through a life cycle of potentially being in the water, animal drinks out of the water, deposits it back in, through feces to the soil. Soil, maybe, heavy rain gets run off into the water again. And we've just got this life cycle of salmonella sort of surviving in the environment, which makes it really challenging. And we know from prior produce safety or food safety programs that contamination really can occur at any point, especially because salmonella can hang around, right, or survive in these non-host environments.
So let's dive in a little bit deeper about Salmonella Newport and Delmarva. Can you sort of explain to us why when we think about Delmarva, we have to think about Salmonella Newport in terms of food safety?
This is a story that just writes itself, right? I keep thinking that one day maybe they'll ask me to be in a movie, a television made movie. Salmonella Newport. So Newport is a specific serovar or type of Salmonella. We didn't really get into that, but there are several types of Salmonella, and they used to be named after kind of the location that they were found, and now we don't do that anymore. We're not as fun. But Salmonella Newport first kind of popped onto the scene in 2002. We had our first outbreak, and we weren't exactly sure where the source of it was coming from. But we were quickly all going to become very intimately familiar with Salmonella Newport and the Delmarva, because over the next series of years, we had repeated number of outbreaks: 2002, 2005, 2007, 2010, 2014. You can see you've got some other possible ones on there that they think might have come from the Delmarva, but were never actually confirmed. This was a huge problem. People were getting sick from eating these tomatoes. They were all being traced back to tomatoes that were being grown on the Delmarva. Specifically, most of these outbreaks were from the eastern shore of Virginia, which was that southernmost part, some of the most narrow part of the Delmarva. So you have that real big special kind of ecological niche between the water and the land and all this stuff going on with agriculture and poultry operations and lots of things going on, right? The people are there. The interesting thing about this is it was the same fingerprint. So we do a lot of fancy molecular type of using tools to sort of figure out like which Salmonella, right? Like which person, right? Let's figure it out. Sleuthing. And we started off with pulse field gel electrophoresis, which is a banding pattern. We've gotten more sophisticated now with technology, right? Now we've got crazy Oculuses and things like that. You can play immersive video game experiences. Now we've got whole genome sequencing, which gives you, this whole picture of that organism. We know that all these organisms from these outbreaks are basically the same match. So it's saying that this Salmonella Newport specific strain, bad actor, as I like to call it, is basically somehow surviving or proliferating on this Delmarva area. And that if we don't have sort of all of our hurdles or our mitigations in place for food safety, we can potentially have problems. And we certainly in the past, knock on wood, we have had our issues. We have not had a confirmed outbreak since 2014 linked to the Delmarva, and we're really quite proud of that. I'm sure we're going to talk about it. But as I said, mostly tomatoes, but we did see some cucumbers in 2014, and I think we're going to talk about that. Figure out how we can prevent it. And so one of the things that a lot of people did with the FDA, CDC, our Virginia Department of Health, Maryland's Department of Health, our Department of Agriculture's universities, our extension services, people got out there and worked with the growers to figure out sort of observationally what's going on. And through these intensive assessments, we sort of identified 4 main outcomes that came out of this. 3 of them were water specific, and 1 of them was packing house specific, right? So we'll say 3 of them were kind of based in the field, 1 a little bit further down the chain, again, where you're packing and getting things ready to be shipped out or distributed. We noticed that in terms of a lot of it was around our water quality. What kind of water were we using for irrigation? How were we irrigating? Was it overhead? Was it drip? We know that there are different risk profiles, right, for each, because it might come into contact with the tomato in this case for the outbreaks. What water we were using for our pesticide sprays. Traditionally with a pest program, pesticide application, you like to use the best quality of water, right? So it doesn't interfere with your chemical, but that wasn't always happening. Sometimes
what was the source of water primarily in Virginia when this was happening?
A lot of ponds. Pond water was a big thing. We've got wells, so we don't really have your really sophisticated city setups where you might have a municipality. It's rural, right? A lot of us are on wells, a lot of driven through ponds. Well, we need so much water that we can't really recharge these wells fast enough sometimes to get the water out. So a lot of times what we do is we'll dig these ponds and we'll fill up the ponds with the well water, and then we'll be using the water from the pond. Typically this is kind of works okay. But again, we were using that water for pesticide applications where it was overhead irrigation. So it's coming into contact with those tomatoes, sometimes pretty close to harvest. Traditionally, at least on the Delmarva, a lot of our tomatoes are irrigated with drip. So, that might be a risk reduction strategy, but for our pesticide applications, we're spraying. So that's overhead. So that was one of the first things we noticed, right? Was that particular setup. And that's just I think many places face those similar challenges with, can you use well water? Can you recharge those wells fast enough, especially for the volume or the output that you need to get out for irrigation or some of our water use activities. And so we see ponds used a lot, or these holding ponds. But when we do that, it makes them open to the environment, right?
Yeah.
So add an extra layer of a little bit of risk there, because now our really low, a really good quality well water, or low risk, being open to the environment adds a challenge there, right? So we had to think through that. And
Did you do any studies? I mean, I know several slides ago we talked about how salmonella can live in the environment. Did you guys find salmonella in those ponds?
we did. And we've, even to this day, we've done several studies over the course of 20 years. And we have shown that you can find salmonella in some of these ponds or in some of the sediments, right? Which is the bottom layer, right, of the pond. And so you can imagine if you're irrigating and you're causing a disturbance, potentially salmonella, right, from those sediments where it's kind of dormant, can get into that water, those water columns, and potentially, if it is used in an overhead fashion, could contaminate crops. Not to say that like we have salmonella everywhere, we don't, but
Right.
yeah, we can find it, of course. I think that's fairly normal. You can find it in the environment. So again, it was really the practice of that we were, using this water, but we were using it for an overhead application because of the pesticide use, and that we were using pond water. Really simple fix, right? If we're going to use pesticide application or overhead irrigate, we 100% need to be using water from the well, or water that's in a holding tank that's been treated, or do some sort of chemical treatment, adding maybe a little bit of chlorine or something to that water to make sure we don't have salmonella in there.
So your third bullet point there under your 3 water related practices is one that I think is really interesting, and it talks about lack of backflow devices during water usage. Can you delve into a little bit more about what that means?
Yeah, so I mentioned that we're filling things up, right, from wells to ponds. And so in that particular setup, we noticed that we weren't using a lot of backflow prevention devices, where some of the water from the pond, right, because it's, you taking the water out of the well, right, with a connection to filling up your pond. We didn't have backflow prevention devices. So what was in that open source kind of risky water pond was potentially then now contaminating our well. Our really good quality water. Again, a really easy fix by adding that backflow prevention device. We typically, see this recommendation when people are filling up sprayers because they don't want the chemical hazards, right, to go back into the well. This, of course, was the opposite. We just didn't want the potential microbial contamination. So again, a really easy fix. And again, we didn't really think about it until we started to really have these conversations around observation, working with the growers, talking about why we were doing certain things. And again, a lot of it was that we do use a lot of water, and sometimes you just cannot get the volume of water out of those wells to be 100% on wells. Plus, that's not really, it wasn't working for them, for the volume of the agriculture that they needed to irrigate.
Perfect. So let's talk a little bit about this 2014 outbreak, where it was not the tomatoes. It was actually cucumber. So can you tell us a little bit about that?
Yeah, so this one was, kind of like a whodunit mystery. And of course, everybody's thinking tomatoes, tomatoes, tomatoes. Turned out it was not the tomatoes. And I think this really solidified for us collectively as a group, growers, helpers of growers, right? All of our our allies, that there's clearly something going on in the environment here. So we have to be so careful about just implementing all of our best practices. But this was one of the ones, as I said, it was a cucumber farm. Again, lots of people affected, lots of hospitalizations. And what they were really able to kind of pinpoint here is when they went back to do all the trace back, most of the product was gone. The farm, of course, wasn't packing product anymore. And like most farms, when they're not packing and they're done for the season, you don't really have a lot left to get really good observations. And so we were left having to go through a lot of the records. And that's where we sort of we think we have a good hypothesis of what occurred. But again, we'll talk about that on the next slide.
Yeah, so let's talk a little bit about the hypothesis of what caused that cucumber outbreak, which is a little bit different, I think, than sort of what we were pointing to in all those salmonella outbreaks.
Right.
Or all those tomato outbreaks, sorry. Yeah.
Salmonella, tomato, it's the same, it's the same thing. But yeah, we had pinpointed all of these little things that had, occurred that make sense of why they happened, right? Because of the volume and the production and just the situation. All easy fixes, right? Even with the flumes, to go back to that a little bit, right? We can try to, minimize the water temperature, add a sanitizer, right? All easy fixes. So for this one, this is a little different, right? So we went back. There wasn't a lot to look at because it was post-season, right? So, we didn't get to see product growing in the field, and through all of that work that we had done on the environmental assessments prior, we got to see and work with the growers, right? We got to talk to people and see production and see what was going on. You can learn a lot more that way. Here we had to go to the records. So through the records, what we saw was that they had been applying poultry litter, which again, we mentioned earlier in the talk that potentially salmonella could be in animals, right? Zoonotic pathogen. So birds, chickens, pretty common carrier of it. We know this. That's why we cook our chickens. We don't eat them raw. So they're applying poultry litter, which is a good potential soil nutrient, right? It's good for soil management plans. Here's the problem with that. We're applying our raw poultry litter, and we were applying it and then harvesting before we had really given there enough time to see die off or whatnot. And so that's what we noticed in the records here is if we get into biological soil amendments or manures, we like to not use raw manures. Okay. That would be our best food safety practice. Don't use raw manure. Okay. Can't always do that because a lot of times we make a lot of manure. I just said there were a lot of poultry production. So we got a lot of free manure. It is a very good way to add nutrients back to the soil. So we might want to use that. But then we have to think through, okay, if I apply this raw manure and I might have potentially contaminated my soil, what can I do from a food safety perspective? Well, these rules actually come from National Organic Program and the USDA, their audit programs for good agricultural practices. They say if you're going to use these manures, then you need to give 120 days before you would go in and harvest. That gives ample time, we'll say, to get some sort of reduction of our potential contaminants. And again, it's using the environment to your benefit. So
I think, Laura, I'm gonna move us on to the last slide here and kind of wrap us up. But we've talked through some of these points here. I think the top 5 in particular, but I think Delmarva and this ongoing outbreak offers us or offers growers some really important lessons about things that they can do to really avoid contamination on their operations, regardless of whether they're in Delmarva or elsewhere. So can you just talk us through things that growers can do and what we can learn from this sort of long history and what was learned on the Delmarva Peninsula such that there's not been an outbreak?
Not a problem.
Right?
I say this and it's like, it's really bad luck to do this.
I know, I know.
The collaboration and the cooperation, right, with all of these partnerships, right, everybody involved, right, from government to, you know, federal government, state government, universities, extension, the growers, getting together and coming up and looking at what was going on and kind of coming up with these hypotheses based on what we know from food safety practices. Since 2014, we have been able to not have any issues, and that's a huge win for us. And so it was all the little huge win, right? And again, it was a team effort. It takes a village. You got to get out there and go see it, and you got to have that back and forth partnership.
Thank you so much, Laura and Michelle, for leading this discussion. I just also want to acknowledge that this work is supported by the Food Safety Outreach Program from the USDA National Institute of Food and Agriculture. So thank you again. If you have any questions, please feel free to email Laura Strawn and Michelle Danieluk, and I hope to see you at the next series.